Let us now proceed to the second part of this presentation. A pleasant day to everyone. I'm Jewel Marisupan-Pare, and I will present to you the characterization of bacterial culture in agar slants. So before anything else, let us first define agar slant. So agar slants are commonly used to generate stocks of bacteria. The limited amount of air within the tube and the small opening function to minimize water loss and exposure to outside air. It includes dust and other particles. Therefore, there is a very little chance of contamination or losing the culture due to the medium of drying up. So, examination of agar slant culture must include the following. The first one is the amount of growth. So it can be scanty or slight, wherein it indicates that the sample organism is only present in the initial sample of inoculum. It can also be moderate. It suggests that the half of the culture media is covered by the sampled organism. It can also be abundant, wherein it suggests that the organism occupied the culture media, and it can be none, wherein it indicates that the organism did not grow in the culture media. Next, we have the color of colony. Despite the fact that the colonies differ in color, as you can see in the illustration, white appears to be the most abundant tone in colony formation. This is because of the absorption of spectrum in different molecules. Next, we have the consistency. So it can be butyrous, wherein the consistency is watery and resembles much like a butter, just like in the picture. It can be viscid, wherein this has a sticky or glutinous with a thick layer consistency. It can also be membranous, wherein it has a thin layer and coherent like a cell membrane. It can also be brittle, wherein the consistency appears to be dry or friable. Then we have the optical characteristics. So it can be opaque, wherein it doesn't allow any light to pass through its surface. It can be translucent, wherein it allows very little to light pass through, but not enough to emit the shape or image of the object being used. It can be opalescent, wherein it resembles an opal in color. It can also be iridescent, wherein it changes the color of the reflected light into rainbow colors. It can be fluorescent. It only reflects one color of light. It can be dull, wherein it is not glossy, and glistening, wherein it is glossy. Next, we have the form. So we have 6 kinds of form, or 6 types. So for the first one, we have the arborescent. It is a tree-like growth appearance of colonies. Then it can be beaded, wherein confluent or separated colonies at the half end of inoculation. It can be echinulate. The colonies form a tooth-like pattern or pointed margins. It can be filiform, wherein colonies are separated throughout the line of inoculation. It can be rhizoid, wherein it is irregular in shape or root-like appearance of colony formation. And last one, it can be spreading, wherein colonies form along the line of inoculation. Next, we have their odor. So microbe, Alcalagenis faecalis, its odor is freshly cut apples. Candida albicans, E. Citrobacter frundi, it smells like a dirty sneakers. Clostridium difficile, fecal. Eichenelia corrodens, bleach or cracker. Haemophilus influenzae, wet fur. Pasteurella multocida, pungent. Streptomyces, smells like musty basement, as well as the nocardia. Pseudomonas aeruginosa, wherein its odor is fruity grape-like. And for the last one, we have the medium change. So bacteria modify the color of the medium based on the chemical presence, such as bile, which hydrolyzes esculin and causes the media to darken from light brown to dark brown. So that's all for the characterization of bacterial culture in agar slants. So thank you for listening.
Characterization of bacterial culture in nutrient broth. What is nutrient broth? Basically, the nutrient broth is the nutrient agar that lacked of the solidifying agent. They remain in liquid form at room temperature and are usually used to maintain the stocks of microorganisms. Also, you can enrich your nutrient broth with blood, serum, sugars, and others for special purposes. In general, they are used to grow fastidious organisms, which has very complicated nutritional requirements, meaning it will not grow without specific factors present or in specific conditions. Examination of broth culture must include the following. First, surface growth. It can be none, ring at upper margin adherent to glass, flocculent, which are small masses of growth of various shapes of floating in the culture fluid, pellicle, which is growth in the form of a continuous or uninterrupted sheet over the surface, or membranous, which is a thin coherent growth like a membrane. Second, subsurface growth. It can be none, turbid or cloudy, granular, which are small particles, flaky, which are large particles, or flocculent, which are small masses of various shapes. Third, amount of growth. It might be none, scanty, moderate, or abundant. Fourth, sediment. It can be none, granular or small granules, flocculent, which are small masses of growth of various shapes, flaky, which are separate flakes, or viscid, which is sediment rises as a coherent swirl. And fifth, pigmentation. It can be none, or usually the color is amber, slightly opalescent. However, the dehydrated medium should be homogeneous, free-flowing, and beige in color. The differential media make it easy to distinguish colonies of different bacteria by a change in the color of the colonies or the color of the medium. Thus, color changes are the result of end products created by the interaction of bacterial enzymes with differential substrates in the medium. Here is an example of pigment production in nutrient broth, wherein the test tubes are labeled as B. blanc, 1. 28 degrees Celsius, 2. 30 degrees Celsius, and 3. 37 degrees Celsius. Addition of maltose to nutrient broth enhanced pigment production at 28 degrees Celsius and 30 degrees Celsius, while at 37 degrees Celsius, Serratia marcescens, which is a gram-negative bacillus that occurs naturally in soil and water and produces a red pigment at room temperature, did not show any pigment production in nutrient broth.
Pleasant day to everyone. I'm Christian Muzones, and today I will present to you the characterization of bacterial culture in agar slants. So the first factor in examining of agar slant culture is the amount of growth. Here we have 4 types of amount identification. First, we have the none, which indicates no growth of the bacteria in the agar slant. Next, we have scanty, which indicates that there is minimal growth. Then we have moderate, which suggests that half of the culture media is occupied by the growth of the bacteria. And finally, we have abundant, which indicates that the bacteria occupied the agar slant. Next, we have the form of growth. Specific bacteria can take different forms as they grow in an agar slant culture. So here we have filiform, next we have bidet, next we have papillae, then we have villus, and finally we have arborescent. Finally, we have liquefaction. Liquefaction is the reduction of a gelatin culture medium to the liquid state by enzymes produced by bacteria in a stab culture. So this is used in identifying specific bacteria. So first we have the crateriform, next we have the napiform, then we have the infundibulum, next we have saccate, and finally we have the stratiform. That will be all for our presentation today, and thank you so much for listening.